Hi guys, welcome back to the channel. In this video I will carry on with the respiration topic, and in this video we will cover the Krebs cycle, oxidative phosphorylation, as well as anaerobic respiration and the rest of respiration topic. So let's begin. We'll start with the Krebs cycle. So this comes after the link reaction. So from the link reaction we gained a acetyl-CoA, acetyl coenzyme A, which we know is a 2-carbon compound. So now in the Krebs cycle, what happens is that we have a 4-carbon compound, which is called oxaloacetate, and what it does is it combines with the acetyl coenzyme A, and we form this 6-carbon compound, and the acetyl coenzyme A is just released back, and it can be used in the link reaction once again to form acetyl coenzyme A again. And then we have the 6-carbon compound losing a carbon by releasing a carbon dioxide. So for example, this carbon dioxide is released, and we have only 5 carbons left now. So next, the NAD, there's the enzyme NAD flowing around, and these NAD can be reduced from the 5-carbon compound by oxidizing the 5-carbon compound, releasing hydrogens, and that reduces NAD. So next, what we do is another carbon dioxide is released. So we have a 5-carbon compound, so now another CO2 released, and this leaves us with a 4-carbon compound, as you can see. And the next step is ATP synthesis. So there will be a phosphate, and it will join with a ADP, and it would form ATP. And next we have 2 NAD coenzymes in this step, and both of these will be reduced. So again, the 4-carbon compound is being oxidized, and NAD will be being reduced by gaining those hydrogen. And in this step we also have another coenzyme which is called FAD, and this is also being reduced in this step. So again, we would lose hydrogens from the 4-carbon compound, and this would be used to reduce the FAD. So in this step we have made an ATP, reduced 2 NAD, as well as reduce an FAD. So we are left with a 4-carbon compound, and this has formed the oxaloacetate again. If you remember, this was the starting block, what we started with, which joined with the acetyl coenzyme A to form that 6-carbon compound. So, and this is why it's called a cycle, because we have formed this oxaloacetate again, which will carry on this reaction, and this will be formed again, and the reaction will keep going on. This is why it's a cycle. So now we are in the last step of respiration, or for aerobic respiration, which is called the oxidative phosphorylation. So now we are in the cristae of mitochondria, and these are the folds, the membrane which is surrounding the mitochondria. So what happens is the coenzymes that we have gained, and the reduced coenzymes from glycolysis, the link reaction, as well as the Krebs cycle, we will have at the start. So what happens is, for example, the NADH2, so it can be any reduced coenzyme really, it can be FAD as well. So these reduced coenzymes are oxidized, and they will lose their hydrogen ions and electrons. So we have these electrons as well as these hydrogen ions. So the coenzymes can go back to the glycolysis or link reaction, or the Krebs cycle, and can be reused again, as they are now oxidized, they're not reduced anymore. So the next step is that the electrons, so these two electrons, what we have gained, they will pass down a series of electron carriers on the mitochondrial membrane. So this is the mitochondrial membrane, and there are a series of electron carriers. So this is very similar to photosynthesis, and they pass down in a series of redox reactions. So what this means is, for example, when they move to this, so if we say this is an electron carrier, when the electrons move out from this to the next electron carrier, they would be reducing this electron carrier. But now, because the electrons have moved out from the first electron carrier, this will become oxidized. So that's how they move in a series of redox reactions. So the next step is that as the electrons have passed along these electron carriers, and they have lost energy throughout. So some of the energy can be used to pump the hydrogen ions, which we will see in a minute, but some of the energy is lost as heat. And it's crucial that you know that some of the energy is lost as heat. Not all of the energy is used to pump the H plus ions. So next, what we have is these H plus ions being transported into the intermembrane space. So we have the inner membrane there, we have the outer membrane there, and we have this intermembrane space, and the H plus ions have been transported to this intermembrane. And what happens next is these hydrogen ions would pass back through the inner membrane via a enzyme, which is the ATP synthase enzyme, and they would form ATP. So these hydrogen ions are moving back through the ATP synthase, and as they're moving back, ADP plus phosphate joins in a condensation reaction, forming ATP. So again, this is very similar to photosynthesis. It's again the same chemiosmotic theory. And the final step, what happens is so we have a oxygen flowing around. What happens is the electrons, so these two electrons we had from the coenzymes, as well as the hydrogen ions we have again from the coenzyme, the reduced coenzyme, which was oxidized. So now these combine with the oxygen. So when they combine, the electrons, the hydrogen ions, and the oxygen, they form a water molecule. So these are combining, and basically a water molecule is formed. And what we say is the oxygen is the terminal electron acceptor. This is a key term because like the coenzymes have been accepting those hydrogen ions and those electrons, but oxygen is the last one to accept those electrons. That's why we say that it's the terminal electron acceptor. This is basically when the reaction has finished. So we have formed a H2 molecule, which is, again, if you remember from the respiration equation, it's one of the final products in respiration. So we have formed that product. I'm going to summarize what we have just looked at now. So starting with the Krebs cycle. So Krebs cycle is obviously occurring in the mitochondrial matrix, so inside the mitochondria, and it's important to visualize the Krebs cycle as a cycle because most of the exam questions look into Krebs cycle as a cycle and how it can be affected. So just remember that Krebs cycle just goes on, keeps going on because it's a cycle. And so starting off, so we the usual starting point for this cycle is what the products of the link reaction. So we have gained the acetyl coenzyme A, which is a 2-carbon compound, and that's reacting with the 4-carbon compound oxaloacetate. So we have this oxaloacetate there, and the acetyl coenzyme is coming in, and as they react, the 6-carbon compound is formed, which is called citrate. And remember that the coenzyme A is released back into the link reaction to be used again. And the next step is that the carbon dioxide is released. So we go from a 6-carbon compound, and releasing carbon dioxide just turns it into a 5-carbon compound. And then obviously the NAD is reduced, and so we have NAD, and then it's gaining those hydrogens, and that just forms reduced NAD, or just NADH2. And so we have the 5-carbon compound, and again another CO2 is released, so we have turned into a 4-carbon compound, and then 2 NAD are reduced this time. So 2 of these NAD come and they gain those hydrogens and they're reduced, and we also have another coenzyme FAD being reduced this time as well, the same way as NAD, and we also have ATP forming in a ATP synthesis reaction, so from ADP and PI. And we have formed the 4-carbon compound again, which is the oxaloacetate, and this can react with acetyl coenzyme A, and the reaction can carry on as this is a cycle. So the net production of Krebs cycle per glucose is 4 carbon dioxide to ATP, which is produced by substrate level phosphorylation. We're going to look at that in a bit. 6 reduced NAD, as well as 2 reduced FAD. So now remember that because per glucose the Krebs cycle would occur twice, that's why we multiply the products by 2. So that's why if we have 2 CO2 formed in a Krebs cycle, we say that and there will be 4 CO2 formed per glucose. So now looking at the oxidative phosphorylation, which is the final step of respiration. So remember, this is occurring in the cristae, which is just the membrane of mitochondria. So these folded parts, and it's occurring in there. And what happens first is the reduced coenzyme. So for example, the NADH2, the FADH2 are oxidized and they lose hydrogen. And what happens is the electrons from those hydrogens, so we end up forming hydrogen ions, and we have electrons from them. So the electrons from these hydrogen ions, these hydrogens, which end up forming hydrogen ions, would pass down a series of electron carriers on mitochondrial membrane in a series of redox reactions. And so again, this is the same as photosynthesis, so that chemiosmotic theory. So as they pass along, they would reduce a electron carrier, and they would be oxidizing another electron carrier which they have just moved from. So next is that the electrons lose energy as they pass. So some of that energy is used to pump the hydrogen ions through the inner membrane into the intermembrane space, but some energy is released as heat. And what happens is the hydrogen ions we have in the intermembrane space, that now they can pass back through the inner membrane via ATP synthase, and ATP would be formed from ATP and PI. And the electrons from hydrogen ions combine with oxygen, forming the electrons as well as the protons. So the electrons and the hydrogen ions combine now with oxygen, forming water. And oxygen, remember, is the final electron acceptor, or the terminal electron acceptor. So it's the last thing to accept those electrons in the aerobic respiration reaction. So we will look into anaerobic respiration now, So in anaerobic respiration, only glycolysis occurs. So for example, in aerobic respiration, we had glycolysis, link reaction, and the Krebs cycle, as well as the oxidative phosphorylation. But in anaerobic respiration, because we don't have oxygen, only glycolysis will occur. And what happens is the reduced NAD, so it be oxidized to NAD. So we have the NADH2, which is the reduced NAD, just being oxidized, losing those hydrogens, forming NAD, and those hydrogens are accepted by pyruvate. So when those hydrogens are accepted by the pyruvate, the pyruvate is reduced to lactate. So this is basically the same as lactic acid, and this occurs in animals. So lactic acids do not build up in plants and microorganisms, and they build up in animals as well as humans. And ethanol is built up in plants and microorganisms. So if you remember that if we carry out anaerobic respiration, we have lactic acid forming. So this is what happens. So the pyruvate, which in aerobic respiration would go into the link reaction, it does not go into the link reaction. Rather, it just gains those hydrogens, and it is reduced to lactate. And so when the oxygen is regained, so when the person is able to carry the aerobic respiration, the lactate or the ethanol can be converted back to pyruvate by oxidizing the pyruvate. So it would be losing those hydrogens. Okay, so now looking into the production of ATP in respiration, you should know that the reduced NAD, so the NADH2 in aerobic respiration, would produce 3 ATP. It's not exactly 3 ATP, but it's rounded up, and that's the average. And the reduced FAD, the FADH2, would produce 2 ATP. So these are the numbers required you to know by the exam board. And we end up forming, rounded up, it's on average it can be less than that, 38 ATP in aerobic respiration. So this is because so from glycolysis, link, and Krebs cycle, we get 10 NADH2. So if we are forming 10 NADH2, and if one NADH2 is forming 3 ATP, we can say 10 times 3, which will be 30, and we're forming 2 FADH2, so that'll be 2 times 2, which is 4, and we're forming 2 ATPs directly from glycolysis and Krebs, so we can add that into it, and we end up getting 38 ATP being produced by aerobic respiration. So now comparing that to anaerobic respiration, we only produced 2 ATP per glucose in anaerobic respiration. You can see the huge difference in anaerobic respiration. Aerobic respiration is much more efficient, and that's why we need aerobic respiration to be able to survive. Okay, so looking into the substrate level phosphorylation, so this is when the production of ATP occurs directly from the transfer of phosphate ion, so from the substrate. So there will be a phosphate ion in the substrate to ADP. So ADP plus the phosphate would result in the production of ATP. So as you can see, we have the substrate there containing the phosphate, and we have the ADP, and this ends up forming ATP. So this is directly. So this is not the same as oxidative phosphorylation, which involves the electron carriers. So this is just direct production of ATP. It does not involve any electron carriers, and this is used to produce ATP in glycolysis as well as the Krebs cycle. So this is substrate level phosphorylation. You need to be aware that there are other respiratory substrates which can be used to produce ATP. It's not just glucose which is used. There are other substrate that can be used. So for example, we can have lipids. So lipids can be hydrolyzed into glycerol and fatty acids, and for example the fatty acids can be converted into acetyl coenzyme A, which can enter the Krebs cycle, and then the Krebs cycle can go on. Or we can also have the amino acids. So what usually happens is the amino group is removed from the amino acids. So amino group is that NH2 group. So this is called deamination, and then this can be converted into acetyl coenzyme A, because there are, so for example, there will be two carbons in this, and the acetyl coenzyme A has 2 carbons. So and again, this can enter the Krebs cycle, and aerobic respiration can carry on. So it's not just glucose which can be used for aerobic respiration. There are other respiratory substrates, and you just need to be aware of them. Thank you so much for watching this video. If you did like this video, don't forget to subscribe to see more of these, and you can watch my recent videos by clicking on the links popping up. Thank you.